All right. So welcome everyone. We're so glad that you're here. We're glad that this is happening. We're really appreciative and excited to be working with Melanie and Dominic Delaney, who are in the UK right now. I think it's 8 o'clock, 9 o'clock?
9 o'clock.
9 p.m. They are both molecular biologists with a teaching instinct. So Origami Organelles was started in 2013, is that correct?
Yeah.
And is a labor of love and a way to kind of really share the experience of, building organelles to solidify, learning. That's what their company organization is all about. So we're really excited, and it feels like a very timely opportunity to be sharing and making this connection between our community and the work that they've put into this. We're also joined by Chris Resch, our veteran workshop leader with 14 years or more teaching AP biology at Montgomery. We're going to be the hour we're going to spend together. Dominic and Melanie are going to introduce their website, their catalog of models. We're going to work on building the life cycle of the coronavirus. And then Chris is going to share a little bit of how he's used that with his students, and he's actually going to share some of his student work. And we'll see how it works. We were thinking of doing breakout sessions, and Chris is going to guide us through that, and we'll come back for some Q&A. A few points about kind of like Zoom norming. You're all going to be muted on arrival. If you have questions, please try to put all of your questions in the chat box so that we don't miss any of them. And let's see. If you do not yet have your file to print out and cut of the coronavirus life cycle, please private message Dominic and Melanie. If you have any issue with that, please also comment in the chat. So without further ado, and we are recording, I will hand over to Melanie and Dominic.
All right. Thank you very much, Yadna. It is really lovely for us to be joining you today. This Zoom webinar is a first for us.
Indeed, yeah.
So hopefully the technology will not fail us. So yeah, as Yadna said, we both used to be research scientists, but have pursued a career in seeking out and developing interesting and fun ways to teach science in the classroom. So Origami Organelles are downloadable paper models for teaching science. And we cover topics in biology, chemistry, and earth science. And what you get when you order an Origami Organelle model is what you've received in the email today. You get a file that has the templates in color and black and white. You get the full instructions on how to make the model and also how to do the activity associated with it. And you also get a fully customizable PowerPoint presentation. And once you have the files, they are yours forever to print as often as you like, forever. You can also upload them onto your password protected distance learning platform such as Google Classroom or whatever you use. So that's the way they work. Anything to add about that?
No, that sounds cool.
Okay.
So, yeah.
All right. Well, what I will do now is there's just one comment there, Dominic, if you just check that.
Okay.
Okay. I'm just going to share our website with you. So let's just get that on board here. And let me just add that so we can see it. Okay. Oop. Can everybody see our website? Yeah? Okay. All right. So this is our Origami website, origamiorganelles.com. It's got the usual stuff across the top menu here, FAQs and how to order. I just want to draw your attention to the products tab here. If you click that, it shows the groupings of models that we have. We have over 120 different models, and we have catch-all categories like biology or some that are more specific like genetics. So if I just click on the biology tab, this is what it takes you to. And what you can see here is we've got our coronavirus models are at the top because they're quite popular right now, as you can imagine. We also have models to teach anatomy, biochemistry, and cell biology and genetics. And I just want to highlight this model here, this is our CRISPR model, the molecules that are used to do genome editing. And each page has a couple of pictures of the model, some description about what you can do with the model, the amount of time it takes, and the dimensions. So that's what you get. Also, what you need, and what you will always need, is obviously paper, some scissors, and some tape. So if you haven't cut your models out already, you should have some scissors with you, and you will be needing a little bit of tape. All right, I think that's all I wanted to say about the website. And now, should we show a couple of models just to get a sense of scale?
So let me show a couple of our models. So hello, everyone. So so here's the coronavirus Melanie mentioned a moment ago. Okay, can I hold that? The colours don't come out quite right on our screen, but you should get the idea, hopefully, of the size and so on. And you can see the main features of the virus that's giving us so much problems at the moment in the world. So that's a kind of fun model. And then we've also got, this is a model of the eye. So this was one which we developed a little while ago, and we've got quite a few anatomy models, and they tend to be very popular models. And if you can see, you can sort of see through the eye in this model. So this is kind of a fun way. You can even have different eye colours. Students can choose their own eye colour and stuff like that. So, and of course, you can print them in black and white, as I'm sure some of you have done today. So don't worry about the colour. We tend to show the colour. This is the plant cell that I know Yadna was particularly interested in. So a model of plant cells. So just showing the main organelles, the nucleus, endoplasmic reticulum, the chloroplast, and so on. So these are quite simple models to make as well.
Just to say about the plant cell one, we have kind of an equivalent animal cell and a bacterial cell, which is really good for comparing and contrasting.
And then just finally show another model, the heart. So this is a model that was kind of fun to work out how to make this. So it's a 3D model of the heart with all the major blood vessels, and you can show the cardiac cycle with it and so on. And that's been consistently one of our most popular models as well. So there we go. So that's a few of our models. And then the coronavirus life cycle, I've just take this one down. So this is what we're going to work on today. I mean, mostly as an example to show you how the system kind of works and what you make when you do a model and how you can use it for an activity. So I need to get into, so shall I begin? Mm-hmm. If anyone has any questions about anything, if you can get the PowerPoint open.
Yeah. 1 second.
We're just answering a question, I think.
Spelling badly.
Okay. So let me share the screen. Okay. Okay. So sorry, excuse us. Technical manipulations here. I think we've got the.
I think we're all learning to be very forgiving about any.
Yeah. We've got the window open on the website, have we? Oh God, no, hang on. That's not it. Here we go. I have to
just
close that. Right.
Can you see our PowerPoint now? No. Okay. You have to click.
Okay. Sorry. Forgive my.
How about now? Can you see the PowerPoint? Excellent. Okay.
Brilliant. Okay, great. So, thumbs up. All right. So you have all got this PowerPoint, and just to say, I have edited it. I've removed a couple of things in it, so you have the full version. But this is essentially the same one that would come. Well, has come with the model that you've got. So let me just find the right key to press.
Okay.
Not that one.
Right. Yeah. I don't have the chat window up right now, so maybe you could keep an eye on that for me. Okay, thank you.
Hang on, how am I think I'm not clicked on it, that's the problem. Right, okay, here we go. Okay, so coronavirus life cycle model, this is one we're going to work through with you guys today. So just to give you a little overview of the life cycle, I don't know how familiar this is with you all. So we've got different features shown, and the way we've presented it is you have here where the virus enters the host cell, which is a lung cell. And then it goes through a series of steps, translating the genome, producing messenger RNA, packaging the viruses, and producing the kind of baby viruses at the end of the cycle. So that's how we'll work through it. Okay, so I'll go through. So you should hopefully have everything you need printed out. If you haven't cut it out, feel free to cut while we're talking, and we can, I mean, obviously you can do this later on if you don't have time to do it while we're going through it as well. But so this is the kind of things you need, just the sheets, tape and scissors.
Oh, if I can just interrupt for
Sure.
1 second. So this PowerPoint that Dominic is using is the PowerPoint that comes with the model. And we have taken out a lot of the slides with the writing because Dominic will say all those things, but all the information and the background science is in the PowerPoint that you have. So.
Okay, thanks. Yeah, cool. Okay, so the first thing we're going to do is make the host cell. So we're going to go straight into making the model because I think this is the best way to see what the idea of origami organelles is about. So hopefully you have one of these pre-cut, and this is the,
sorry, could we go back to the slide that shows all of the pieces?
Yes, sure.
Yeah, thanks. I just want to muse on that just for a second. And maybe
Sorry, yeah, sure. I mean, I can talk through it in more detail if you like.
Super quick, maybe?
Yeah, sure. I mean, okay, so no worries. So, so the host, okay, so the virus attaches itself to a particular receptor on the membrane of the host cell, and this is why it targets particular cells. The lung cells have this receptor expressed on the outside of them, and through that receptor, they're able to introduce the RNA genome into the host cell, and then I'll talk through it now, and then we'll go back over it. And then the host cell ribosomes use that genome, which is RNA, they recognize it as a messenger RNA and make proteins from it. And these proteins make these enzymes, which are made, replicate the genome of the virus, all in RNA. There's no DNA involved in any of this. And then the messenger RNAs are made from the viral genome, and then you end up packaging the viral particles. And it goes through the normal parts, the normal organelles in the cell. It goes through the endoplasmic reticulum, the Golgi apparatus, and it comes out in a vesicle, which then fuses with the cell membrane. So it kind of goes through a series of steps. It's quite a nice, interesting, different way of showing transcription, translation, replication of genomes in a completely different context. So it's kind of interesting for students to have to think about it in those terms, I think. So, you know, obviously not everyone learns virology, I guess. But it is a very interesting way. Is that okay? It's like
no, it's great. Thank you.
Okay, cool. Okay, so we'll go through just making the model now. So yeah, just so in the origami organelles world, so when it says solid black lines, it means a cut. So we tend to say cut around solid black lines, and the dashed lines are where we want someone to fold something. So if you just take your template, your host cell membrane, and fold the sides. I'll show it. I imagine my window is very tiny on your screen, but I've got pictures of everything in the PowerPoint. So you just fold along the sides and fold up the tab. Okay, Molly's sort of showing that, if you can see that, hopefully. And then I'll go to the next picture because then it shows it more clearly. So this is what you'll end up with. And then you're going to take that underneath the membrane using the tab. So you take these tabs underneath to create the membrane. And we use similar kind of techniques in different models because it makes it easier if you do a few different models and students. If you, oh, okay. So there we go. So if you do that, and I'll continue talking whilst we're doing that. Okay. So that will be what it looks like. And then you just fold down those proteins so they're sticking out on the outside of the cell, the host cell, as it will become shortly. Okay, and then just to get that, for more parts we need for that, so we make the enzymes. So these are just cutting out the replicase, the protease I'm holding there, and the replicase, you're just cutting those out. You don't have to do any folding with those. And then with the genomes and the actual messenger RNA and the proteins, again, just cut them out. So they're just strips of paper. Okay. So hopefully you've got some of these things ready. The Golgi apparatus, so I've got, this is the cutout version, so I'll find the camera. And then if you fold the tabs over the back, and then you can just tape that together. So there's Melanie showing that for you. Okay, right. Okay. And then the virus particles, so these are, I say we'll have a go through it and then you can kind of catch as we go through. So this is the viral particles cut out. And then with this one, you just fold it with the writing on the inside into a circle, make a hoop, and then you tape it together. I don't know, we're going quite quickly here. Maybe we have to. Is everyone doing stuff? Are we okay? Do you want me to slow down? Are we okay?
You can also show off your progress every now and then, hold up where you are to the screen. Everybody's, I can see, scissoring away.
Right, okay. Ah, okay. So I can see people working. This is great. Okay.
Maybe try to turn something.
Okay, we're just trying to. Right. Okay. So we are. So where I'm up to is here. I make my model as we go. Once you've made those parts, you just go on. Okay, so that's the virus particle. And then to finish the virus particle, you just have to insert the genome inside. So we've got a couple of different versions of the genome. So what you're looking for is the sense genome. And we'll just fold that into the viral particle, into the membrane, and that will make that join together. So it goes inside. Hopefully it will escape from me.
Has anybody got some finished products for us to see? Do you just want to hold them up to the screen?
How are we getting on?
Lovely.
Beautiful. Missy Golgies. Okay.
You can click to some of the people.
Okay. All right. Shall I continue on? Okay. Sorry. Technical. Okay. So the ribosome requires cutting out, if you haven't already done so. I can't see myself now. Let me just go back a step. Okay, so here's the ribosome. So you've got to make 2 of these, and you cut it out, and then you just fold it around and tape it together as that. And there's one we prepared earlier, as they say. So that's the ribosome. So yeah, and the ribosome has it just shows the various bits, the subunits of the ribosome. I mean, again, you can cover different things with this, so it does actually show that large and small subunits of the ribosome, even if it's in very small form. And then the last thing we're going to make is the vesicle, which we'll need in a few minutes. But to say, we can just go through it, and then you can catch up with us. So the vesicle is here, and we're going to fold up the sides and the dashed line and curl the sides around and tape them together, and that will make the vesicle. Okay, so let me just use my PowerPoint to show. So that's what you should end up with. So let's give you a moment or two to catch up if you haven't done so already, and then we'll move on with that. I can see people busying away. This is good. Some people say I find it quite relaxing when they make these models. So hopefully you're finding that today. And I can start talking through the actual life cycle, because we'll be using the model for that. Okay, so just carry on while we're working on. So we're going to use the first part you made, we're going to use in a moment. And that is the cell membrane from the host cell. And you just need to put your ribosomes and the Golgi apparatus in there. And then that will be what you infect with the viral particle. Okay, how's everyone getting on? Are you okay in terms of the making the model? Anyone need me to hold on, or are we okay? Okay.
Oh, very nice.
Very good, very good. Okay, cool. All right, I'll keep going. Okay, so as I said earlier on, so coronavirus uses this protein that sticks out called the spike protein, and this is what attaches to the receptor on the cell, and it's this ACE2 receptor, which is used nothing to do with the virus, and but the virus uses this as a way of entering the cell. So with your model, once you've got your model, you can try this out. And then we've got questions, so I'll just talk about this a little bit. So what through the PowerPoint, I have taken some of them out just for time, but the receptor questions for the students to think about. So for instance, is the receptor protein for the virus or is it for something else? And of course, the cell doesn't have it on the surface for the virus to use. It's for something else. So that's just one of the questions. And then the other one I've got on here is, given that it causes, what type of cells might have this receptor? And again, obviously this is the lung cells that express this most highly. And this is why this particular virus is very dangerous, because it attacks the lung cells directly. It doesn't attack in the throat or elsewhere. So it goes straight into the lungs, and this is why it causes so many problems for people. So the interaction between the receptor and the spike protein is what allows the virus to insert its genome into the cell. And again, this is another thing you can use to talk about topics like it uses perhaps membrane fusion or endocytosis, where a vesicle is taken inside the host cell. I don't think it's actually really well understood how it works in this particular coronavirus. But anyway, at this point, the viral genome gets inside the host cell. I mean, borrow one of yours, yeah. Okay, so if you just put that there, that's essentially where we're at. Okay. And then the next stage, so the host cell ribosomes will immediately begin to translate that genome because they recognize it as a messenger RNA. So it has 5' methylated end and it has a poly A tail. So the cell thinks it's a messenger RNA. So it makes a protein, a polyprotein, and it also makes a protease. And the polyprotein has 16 parts to it, which we've represented with banding on our piece of paper. And it's cut up by the protease. And this then forms something called replicase. And replicase is an enzyme complex that the virus then uses for everything else in its life cycle. Okay, so the replicase is a key drug target because it's unique to the virus and not something that would normally be existing in our cells. Okay, so replicase immediately makes an antisense copy of the viral genome. So it copies its own genome, but makes an antisense copy. And so you get double-stranded RNA in the cell. And this is, the reason this is done, again, there's a question on there, is because this is used as a template to make more copies of the genome, more sense copies. And that's what it begins to do next. It then starts to use the antisense as a template for the sense. So you can put these pieces in as we're going through the model, and this is what the students will see. And we'll see that when Chris shows the work from his students. So I'm kind of keen that we get to that. And then the next stage is replicase makes shorter transcripts of the genome and then messenger RNAs. And so these are for the structural proteins of the virus. So this is the spike protein and the other proteins that it uses to carry out its life cycle. And so these are translated again by host cell ribosomes. So again, as all viruses, it's parasitic. It doesn't have its own ribosome, so it uses the ribosomes of the host cell. And it makes the thing I showed you earlier, and it puts the membrane into a membrane. It puts these proteins, the spike proteins and so on. And this all happens in the endoplasmic reticulum. And then the virus, so then all you need to do now, what happens in terms of the model is you just add the sense genome, one of the sense genomes, into the viral particle you've just made, the membrane. And then that becomes a viral particle, and it goes through the Golgi and comes out in a vesicle. So this is where you use your vesicle from earlier on. It's a camera. And that's how the virus remains until it's budded off from the host cell.
And if I could just add here, with enveloped viruses, usually an enveloped virus will catch its viral proteins on the plasma membrane of the cell and bud out through the plasma membrane. What's unique about coronaviruses is that they pick up their membrane from the Golgi apparatus and they get their viral proteins there, not on the cell surface, which is quite a difference.
So yeah. So and then the final stage is the virus particles are released. So this is when it gets out into the environment. So obviously it could go into another host cell, into another cell in the lungs, or it can get into a droplet and be coughed out or breathed out by someone who has the virus and infect another person. So that's kind of the end of the life cycle. So just to recap, so we started off with, I'll just use the cursor, we started off with it entering the host cell, you can see me pointing, and then the genome was translated, so we've got translation. Protease then digested the polyprotein that's produced, and it turned it into replicase, and some of the host cell proteins are involved in that. And then it made an antisense genome, which became the template to make the sense genome. And then some of the replicase also makes shorter copies of parts of the genome, which are messenger RNAs, and these are used to make proteins that are in the membrane of the virus. And then they're packaged into a membrane derived from the endoplasmic reticulum, and then assembled into viral particles, and then end up in a vesicle and released. And so that's the whole life cycle. So it's sort of, so what we hope you can see with this model is, although it's obviously just a paper model, but it actually shows you can teach quite complex concepts using these types of models. And this is kind of what we're hoping people will see from today's session, that you can actually use the models, you know, to do quite complex things, and in quite a sustainable way, hopefully, because it is just paper, so it's quite a nice resource that you're using. And so that's where I'm at in terms of, that's where we're at in terms of the model. So I just have a congratulations slide at the end always. And then what I was going to suggest now is we hand over to Chris, because he's got some stuff from students and used this in his lessons, and I think it'll be really interesting for people to see what he's done with his students. So.
Yeah, I don't know if congratulations is in order for making coronavirus, but
very true,
but congratulations for definitely making. I made a Golgi. I got part of it. Here we go. I got part of it, and just so if folks were interested, when we were doing sort of our dry run, we did the animal cell. So I kind of have that one.
That's very nice, Chris. Good work.
Thank you. The folding was shaky for a little bit, but woohoo, nice. I saw a couple. Yeah, so one of the things that, for me, how I found out about Origami Organelles was primarily by accidents, actually. So our school district, just to kind of give you the backstory, went on distance learning rather suddenly. So we had about a day or so to actually kind of get some plans together and things like that. And it just so happened that we happened to be on viruses. So this is with my AP biology classes. And so I was trying to figure out a way to kind of achieve this distance learning, but also be able to see and make the thinking of my students visible. So how is it that, in a distance learning setting, can I actually see what the kids are thinking and doing and learning and stuff like that? So it just kind of, like I said, it just kind of stumbled in my lap that I found this Origami Organelle website, and we just happened to be talking about viruses right as we started distance learning. And it's like, oh, cool, very nice. It seems to go really well together. This might be a fun way for the students to kind of demonstrate their thinking, demonstrate what they know about how viruses work, and their life cycle of viruses in this distance learning setting. And it's not just another kind of electronic video or something along those lines. It kind of gets them away from their devices a little bit. I think a healthy thing right now. So I'll share with you my screen. Let's see here. Can you folks see that?
Yeah.
Yeah.
So at the time we started distance learning in March, like I said, we were talking about viruses. And so they had just completed a bunch of work and readings and things like that on the virus life cycles in general and things like that. And so I challenged my students to come up with a 3-minute video of a virus life cycle using the origami for coronavirus. Very timely, perfect. It kind of fell right into the lap in terms of timing and things like that. So I have 2, and that's literally the prompts that I have on the screen. That is the prompt I gave them, just to create a 3-minute video about how coronavirus infects a lung cell. So the next 2 videos are some of what the students started to produce with these. So I'll play these, and then we can sort of talk a little bit about them afterwards. Just make sure I got that up there.
Here is a host lung cell. The Golgi body and ribosomes are used by the coronavirus to replicate and release.
Can folks hear the audio?
In the coronavirus is a sense genome. The sense genome is a single strand of mRNA which can encode for a protein. The spike protein attaches to the lung cell receptor and sense genome goes through. The unsuspecting ribosome translates the viral mRNA to produce a polyprotein and a protease. The protease cuts the polyprotein up, and the chunks of the protein form replicase, which is used to create an antisense genome. The antisense genome is a non-coding DNA strand which the replicase uses to make more sense genomes and shortened replicas of the antisense, which act as mRNA coding to produce structural components of the virus like the various external proteins and envelope for the virus. The lung cell ribosome translates the short mRNA to produce the structure of the virus. The structure and reproduced sense genome combine as it goes through the Golgi body and enters a vesicle. External spike proteins are already on the outer side of the lung cell membrane, and the vesicle fuses with the membrane, allowing the newly replicated coronavirus to leave the cell. The virus can quickly replicate and invade the host.
And then the end. Yeah. So that was just one example. I tried to pick 2 examples of just kind of how students approach this differently. So these students decided to do or this student I should say, because it was an individual, decided to do this by stop animation. Others, like this student, decided to do it a little bit differently, and I'll play this one for you.
It's a lung cell. Unbeknownst to this cell, it will be infected by a coronavirus, and there will be no RNAi to save it in this origami demonstration. For simplification, the cell only has the Golgi body and the ribosomes depicted, which will be removed until they are necessary for further simplification. The first event in the process is the coronavirus phage coming in contact with the cell. The spike proteins, the orange tabs on the outside of the virus, attach to the host cell receptor, the blue tab, and enter the host cell. Here, a host of proteins on the surface of the virus aid in the process of invading the cell. Hemagglutinin esterase dimer, or the HE protein, aids this attachment process. The membrane protein, or the M protein, aids in the shaping of the envelope later. The envelope protein, or the E protein, is a very small protein which interacts with the M protein in the forming of the envelope. However, on this model, only the spike proteins are depicted. When the virus enters the cell, it releases its genome. Here, there are either 2 paths that the virus will take depending on external factors, which is a property of the coronavirus as well as other viruses. One is the lysogenic cycle, which will bring the genome of the virus into the nucleus and add it to the cell genome. When the cell reproduces as normal, it will make copies of its own genome along with the genome of the virus. The virus here is very sneaky as it can hide in the host cell genome for many generations. It stays with the cells until other external factors dictate whether the virus in the cell now moves to the lytic cycle. In the lytic cycle, the virus will reproduce many times within the cell until they break out of the cell, lysing it, hence the name lytic cycle. Keep in mind there will be much more than 2 viruses created in this process. In the lytic cycle, the viral genome is split into the antisense, or non-coding, and sense, or coding genomes. The sense genome is replicated as well as translated into a polyprotein and the viral protease. The viral protease is an enzyme to catalyze reactions, which, in this case, is to cut up the polyprotein created, which will form the replicase. The replicase for all types of coronavirus is now able to replicate RNA using the RNA template, which is the antisense genome from the original virus, like the transcription process. Through mRNA processing, the mRNA is shortened because the introns are removed, as well as other parts of the mRNA processing, such as the 5' cap addition, as well as any cleavage of the mRNA. Then, the mRNA leaves the nucleus and is translated by the ribosomes into the proteins, known as the capsid, which will later encapsulate the viral DNA, which includes the spike proteins, which are represented by the orange tabs on the outside of the host cell. The coding genome of the virus, the sense genome, is encapsulated within the viral protein, or the capsid, which was translated from the mRNA. The entire protein DNA combo is then moved to the Golgi body, where they are shipped out of the cell within a vesicle. The vesicle from the Golgi body merges with the host cell membrane, and as the virus now passes through, effectively lysing the cell and creating the viruses. Keep in mind that millions of viruses can be created, and the process will restart as the new viruses spread to other cells. And that is the complete life cycle of the coronavirus within a lung cell. All types of coronavirus, including SARS and MERS, have lipid membranes. Soap, which is amphiphilic, can create bonds with the cell membranes in the water, and with enough scrubbing, like 20 seconds of it, can rip the virus apart effectively, preventing it from continuing to spread.
So, what was that?
Fantastic.
Yeah, so those were just two kind of examples, and many of the students kind of went to town with it. And so when I started, I kind of asked some students afterwards, so what did you think, and how would you like the process of going through the origami? And they actually really enjoyed it. One student said they found it extremely soothing, and it was nice to get away from their computer and their parents and sibling and to lock themselves in a room just to record. And so the response was positive enough that we actually ended up doing a second assignment with this. So we actually, they're working on an assignment about cell division and cell cycles and stuff like that, and are doing the same kind of assignment where they're taking some of these origamis, they're making stop motions, or in this case, like this student did just a standard kind of video with the fast scribbling. I don't know what it's called, but that's what he did, and they're getting very creative with it. So for me, I mean, this was a great way to be able to see kind of what the students knew at the end of talking about the lytic and the lysogenic cycle and how do viruses work and reproduce. And there's a lot of evidence of student thinking and student understanding from doing these things. So for me, I know this is my AP students, and some may be thinking, holy smokes, I know there's elementary school teachers in the crowd, as well as middle school and high school, and not everybody is teaching AP. So I was kind of hoping to open this up a little bit. How could folks sort of foresee using this as a way of accessing some of that student thinking and some of that student understanding, especially if given the challenges of a distance learning sort of thing. Is there another sort of origami that's out there on the web pages that might be appropriate for you if you're teaching elementary school? Is there an origami on the web page, and you can kind of just surf on as we're talking, that might be appropriate for you, that you might find useful, that can fit into the curriculum, that might be able to actually do this, to show that student thinking, to put that onto display. So originally, I think what we were going to do was to break it out by grade band. But since there is, how many do we have on the call?
We have 17 total. Thanks to the folks who changed your screening. Did you freeze? Oh no.
It's frozen.
Oh, there you go.
Oh, Okay, all of a sudden everything went black and I started to have a panic attack. So are we okay just kind of talking as one big group? Or would we rather split into two small? You tell me how you wanna, if you wanna discuss and then come back.
Everyone, you can unmute yourself.
I'm okay talking in a group. That's fine.
Okay.
Same
Okay.
here.
Yeah, all right. So let's stay as one big group then. You know, so in terms of kind of visioning for your students, how could you possibly foresee using this to access that student thinking? This was just the videos were just the way that I did it, but I'm sure there's certainly lots of other ideas that are floating around inside everybody's heads right now.
So I'm looking on the website and I see that there's an estuaries one, and even though I teach 4th grade and not AP Bio, I'm thinking that for our Trout in the Classroom project, where we discuss watersheds and trout habitats, this could be a really great material to use to support their learning.
So what, if you don't mind me asking, what kind of assignment are you thinking that your students might be able to do with this?
I think that when they're thinking about trout habitat and how trout habitat affects the growth and survival of the trout, having a tangible model in front of them that they could understand, you know, in this ecosystem, what are the living and non-living parts that are interacting? What does this have to do with the trout? How does this affect the trout positively, negatively? Being able to actually touch and point to these different elements is going to be, I think, a lot easier in kind of like a 3D way than, you know, what we've been doing, you know, 2D diagrams in the past.
So being able to physically manipulate it and kind of explain what's going on as they're moving stuff around.
Yeah.
Is sort of where the vision's at for you right now?
Exactly.
Cool. Are there any other elementary school folks out there that are taking a look at the website and have some other ideas like that?
I'm elementary, but I'm having technical difficulties. Hey.
Uh-oh. I think we're all having technical. Yeah.
I feel like my life is a technical difficulty. But I'm working on it. But look, I got this. I got a lot of this done.
Oh, very nice. Very nice. What grades do you teach?
I teach 4th grade as well.
Okay.
And before I had to restart this browser, I saw that there were also like elementary school links as well. I saw the estuary, and there is also. I mean, we don't talk about animal cells though, like in 4th grade. But it's also interesting. Like there are students who are really interested in advanced topics in biology as well. So I could also imagine seeing like sort of some interest centers being set up that sort of capitalize on students' personal interests as well.
So by interest center, you mean like, here's a spot where they go and they can build.
Yeah, I was actually still thinking about my classroom. But I guess digitally I could like assign enrichment tasks as well.
Yeah. No, I think it's great you're thinking about it in the classroom too, because that's something my colleague, when I originally pitched this idea to my colleague for our AP biology students, I was like, I'm thinking we should do this in the classroom too. Why not? And let's keep going with it.
And someday we make it back.
Yeah, whenever we make it back in the classroom. Yeah, absolutely. I like that idea of interest centers. That's a really neat idea. So students would be able to kind of go over there and look at maybe something like the simple animal cell. I know there's a model on the website for that too.
Can I add, I think this is actually related to something that Chris, you had shared during our planning session, that this could also be used as a part of as an assessment piece or a piece of assessment. And, you know, there's like the planning that the students have to do that leads up to this, you know, visual output. And that can I suppose that would vary by student or by class, by grade.
Mm-hmm.
Just thinking about fourth grade specifically, one of the performance expectations for NGSS for fourth grade is develop a model to describe that light reflecting from objects and entering the eye allows objects to be seen. And in the past, I used to have my kids do clay models or draw their own models, but I could see the origami model being a really great tool for use with that performance expectation.
Cool.
Just that's an interesting point. The eye, we actually have two versions of the eye. The one that Dominic showed has 3 layers in it, but we also have a simpler version of that one, don't we?
Yeah, that would certainly be more suitable for fourth grade. But yeah, I mean, it would, the same idea that it has you can shine a light through it and so on, but it's just a simpler model to make. So yeah, I mean, we've been trying to develop more models for sort of elementary and middle school ages. We sort of started out, I suppose, at the, I don't know, high school level certainly, and then we've kind of been trying to broaden out. So any ideas, obviously, I think Chris is at the beginning, if anyone has any ideas of models that would be interesting, or adapting ones. You mentioned the estuaries one. I mean, obviously, you know, we could tweak that to be relevant in terms of species and so on to different, because at the moment it's, you know, set in a particular place, so we could easily adapt that. Or it'd be interesting to hear if that kind of stuff would be interesting.
Yeah. How about middle school folks? Are there any middle school folks here? Or folks that work with middle schoolers? I know not everybody's in the classroom, but could you foresee middle school folks? Could you foresee using something like this with your students, either in the distance learning setting or in a classroom setting as a way to sort of access that student thinking?
I see a lot of high school folks.
Yeah, I'm thinking we might not have too many. Yeah, middle. Let's shift it to high school then.
I signed up for this thinking about my new classes that I'm going to be teaching next year. Well, this year I taught a biochemistry class for the first time, and now I wish I would have had this when we were doing glycolysis and fermentation and all those cycles, because I did have kids who made videos, but it would have been really helpful for them to have the origami aspect to it and already created, and then they could just concentrate on showing their knowledge, not necessarily drawing the right thing. But also I'll be teaching microbio this next year for the first time. So very interested in doing this, especially if we're going to be in cycles of school at home or whatever, which is possible.
Yeah, everything is possible at this moment.
Yep. I'm already planning for it just in case.
Yeah.
It's just to say, I mean, it's interesting because in the UK it's exactly the same at the moment. So schools are waiting to see, are they going to go back? Are they not? Will they go back after the summer, or whether it will be before? And they cancel all the exams. It's just very, very uncharted waters, really.
Chaotic. And I think as parents, when we've been getting work for, we've got kids in elementary school and one just at the beginning of secondary school, when we've been getting work to go through with them, it's worksheet after worksheet after worksheet, and they get a bit bored. So ours are getting some origami in the afternoon, you know, which they're really enjoying. And our 7-year-old loved making his animal cell. He's quite switched on to that. But I think that's something that is really helpful to do something different, just because it is otherwise it is staring at a screen all, you know.
Yeah, there's only so many videos and so many worksheets and packets and whatever it is that can be sent home. Like to have the kids sort of interact with something is what kind of drew me to it and to assigning it electronically. But how about other folks? Could you foresee using something like this in your classrooms, or virtual or otherwise?
I mean, one thing that I thought of with my, because I'm doing an infectious disease unit with my students, and thinking about if they learn this way that the coronavirus operates, they're learning about different viruses and having them show how different viruses operate within the cell. Like, because I think because I have assigned my students to make their own coronavirus, but I imagine that for some it's really overwhelming to think of like, well, how am I supposed to actually show this? And so what's nice about this is you have a template that kind of can simply show that this is what this virus looks like, and then I think the kids they have an understanding of how other viruses may not have membranes. They would show that differently, and maybe showing like that some viruses would leave and get the receptors as they leave, and it has nothing to do with the Golgi apparatus. Like, I think they could show different scenarios depending on the virus once they already have a template kind of started.
Mm-hmm. Yeah, it kind of gives them sort of like a baseline for where to start, is what I'm hearing from you, Carrie.
Yeah, I think so. And I mean, I was planning to use it much the same way that you had with the video, because I think I do that with different, with claymation type videos, and the kids get a lot out of it. So, and I was interested in, for the same reason, just something hands-on. A lot of students are complaining about just sitting all day long. So.
Yeah. It can get quite grueling for them on the flip side of things if all they're doing is worksheets and watching videos and things like that. And some things that I found, with this is some students that didn't have a printer or something like that actually ended up drawing it themselves. So even though they couldn't print it, they kind of used the images as a template to be able to then, or a scaffold, to be able to then describe how the virus life cycle works. And more recently, like I was saying, with cell division and cell signaling and things like that. And then students get into it and they start adding other props. We've had many cameos of siblings and parents coming in and trying to explain things and stuff like that. So I've found a lot of success with it. And especially, if as teachers we're sort of valuing this ability to access student thinking and to really get in, to have students do the explaining and to kind of do the heavy lifting for a lot of it. I've found this a useful way to be able to do that, especially in a distance learning setting, even if it's something basic or something a little more complicated. So after we did the coronavirus, I think I went on their website and ordered like a whole bunch of extra for the rest of the school year. And even started thinking a little bit about how do you blow them up so that you can get multiple students when we're back in the classroom, so you can get multiple students that are working on one model as opposed to just one student per model. So then they can have that interaction and that discussion.
Or if students had different parts of like the body and then they had to connect them and show how they work from, one part or one cell to the next, that would be cool too.
Yeah, that would be really cool. Somebody has the esophagus and the stomach and, the small intestines and things. Yeah, absolutely.
They could do it like Brady Bunch style,
Really?
So where are folks at? Do we have coronavirus models done? I've got part of mine done.
Everybody show off what you've created.
Everybody show off. Whoa.
Beautiful.
Look at that.
We're
Can we get a screenshot of everybody like holding up their...
Oh, you gotta end your screen share.
That's me. Hold on. How do I do this?
We are getting to the end of the hour. I was wondering if maybe Melanie and Dominic, there are a couple of questions in the chat box we can answer before we say goodbye.
Yeah.
Everybody also hold up your
if you no matter what stage you're at with your even if it's just printed and you just want to hold that up.
Excellent, thank you.
That meeting.
Bashful
Nice.
Coronavirus lifestyle. So we have
the questions. Shall we answer a couple of questions? So yeah, one of the questions was, can you say why it's so contagious to humans. And I think coronaviruses generally, I think, are very contagious because of they're big viruses, and they have a lot of structure to them. So I think they survive quite well outside of the body, which is where this whole discussion goes on about how long, you know, everyone's considering wearing gloves and all this kind of stuff. So I think this is why, but I am not a virologist. I wouldn't claim to be in any way, but I think that must be the reason, because they're very, very sophisticated viruses, which is unfortunately what makes them so good at what they do, which isn't helping us at the moment. So yeah, that was one.
Can I ask this question? I'm an elementary school teacher. I have not talked about viruses since I was in high school. So like, I really do not remember this that well. And I don't, like, I understand viruses do this. They like make the cells replicate their DNA. I remember that. But like, why, like, you know, why is this so much more lethal? Or is it? Like, I don't understand. Why are we doing this right now, but we don't do this for the flu?
Yeah.
Well,
I don't know that anyone knows, really.
Okay.
No, well,
I don't know what it is.
I think that's a really great question. To some degree, it has to do with the overproduction of cytokines at one point, and that happens localized to lung tissue. There are really good research articles out there, and we can definitely put a few of those together.
Okay, I think
Harry has a good response for that.
Well, I think part of it from what I was reading is that depending on the patient, it could be deadly, but to others it's not. And then that combined with you don't know if someone's sick and they're infecting people a lot is part of it. Like if it were extremely deadly, then the hosts would be gone and then it could stop more easily. But it's kind of like a little bit of both, and that kind of keeps it progressing. And I think it's which tissue that it invades, that it's invading lung tissue as opposed to, I don't know, some other organ that you need. It's I think that's part of it too. It's a little bit of everything.
I think I read somewhere just the other day that folks that had, and anybody please correct me if I'm wrong about this, folks that have actually a stronger immune system tend to fare worse than those that have a weaker immune system, because
the cytokines.
Yeah, because of the cytokine release. So if you have a stronger immune system, you release more of these cytokines, and then you have more inflammation. And so the result is all the problems that go along with it, like, you know, fluid collection in the lungs and those kinds of symptoms. So folks that you know don't have a weaker immune system, that don't produce as much of those cytokines, actually fare better, in part because they don't have those side effects as a result of the immune response, which is in part also too why I think they are warning against ibuprofen and other anti-inflammatories, because then it kind of hides the virus a little bit in your system, and you can easily spread it to other individuals.
Yeah, there's also issues of viral load and multiple strains, unfortunately.
Yeah.
Well, okay, so on that note, well, you know, it's specific to the model that we did build, so
very relevant. Yeah.
All really important questions. We're saving the chat, so any questions, I think maybe we can find a way to follow up. If you have any residual questions to send me to connect you directly with Dominic and Melanie. If there aren't any further questions, just to be respectful of time, we're so happy to see everybody and to see everybody working and snipping and taping and just engaging and just being here all in the same room virtually. I want to give one extra thanks to Chris and also to Melanie and Dominic for coming together. So virtual high five.
Thank you so much for having us. This has been really, really interesting and fun to meet you all. So thank you.
Yeah, it's been fabulous.
Unmute yourself.
It's been great.
Yeah. And say thanks. Thanks, everybody.
Thank you so much. Thank you.
Thank you.
Thank you. Bye-bye.
Bye.
Bye.
Bye. Bye-bye.
Okay.